Hello, everyone. My name is Will Stonehouse, and I'm a cognitive science major at UCLA, conducting research at the Ristman Memory Lab. As a cognitive scientist, I'm interested in memory as it relates to brain activity. So today we'll be talking to you about how we can better understand working memory capacity by fusing fMRI and EEG data. So think back to when you last really need to use the bathroom, but you're actually in a Starbucks and you need a passcode to get in. So you run to the barista reading out names to ask for the 5-digit password. Why is it 5 instead of 4? Who knows? It just is. And you try to hold on to it for as long as it takes you to get to the door. So you hold on to it, you finally get to the door, you have to recall the information and punch it in. Well, when you do that, you're actually using a cognitive system called working memory. And as you can probably guess, it has a highly limited capacity. People with greater working memory capacity have advantages in many settings, such as tests of reasoning, comprehension, standardized tests, multitasking, and more. And this supports the idea of working memory as a highly dynamic information processor. And as cognitive neuroscientists, we're interested in the relationship between a construct like working memory capacity and brain activity. And we can do this by varying the cognitive load or the job demands on the brain and have you perform a task in the same way you did with your Starbucks experience. So this is a delayed recognition task where you encode the face, maintain it for a bit, and then retrieve them. Doesn't it seem intuitive that holding more information would require more work? It would also seem more intuitive that the better working memory capacity an individual has, the more brain activity. Research from my lab found that that the regions sensitive to this extra work that you have to put in come from executive function regions. And despite our intuition that this would be a linear relationship, our lab's research found that there's an inverted U relationship between working memory capacity and these load effects, meaning that individuals in the higher working memory capacity right here required less cognitive demand while maintaining information. Remember that maintaining information is when you don't have access to the stimulus. So we think that they're doing something here in the encoding period that gives them an advantage in the maintenance period. So the hypothesis or the research question is, why do high working memory capacity subjects require less cognitive effort while maintaining information? Well, we think that high working memory capacity subjects have better encoding processes. And if that's the case, we should be able to replicate the inverted U relationships that are seen in the maintenance period, particularly in the frontoparietal network. And we also expect to find a linear relationship at the encoding process um, among working memory capacity during the encoding period. So um, I am doing a multimodal analysis. This is advantageous because it uses the best parts of fMRI and the best parts of EEG. Imagine fMRI has a spatial resolution of millimeters, while EEG has an awful one of inches. And then fMRI has an awful resolution, temporal resolution in seconds, while EEG has an amazing one, can take about 500 samples in a second. So by taking the best of both worlds and fusing them, this analysis takes an extremely precise look into what's going on in your brain. Another advantage to the multimodal method is that there are two key components in the EEG literature with respect to visual processing and cognitive workload. It's the N170 waveform that is elicited when you see a face, and the P3B waveform that is elicited when you have cognitive workload. So what we're going to do is we're going to look for these two signals that are mixed in with a bunch of noise and a bunch of other signals in the EEG electrode. And so we're going to employ a type of analysis called independent component analysis. The best way for me to explain it is that it's a type of blind source separation. Think about a ton of yarn all mixed in together, a ton of different yarns mixed into one big ball. That is essentially what the EEG signals are. It's a bunch of different waveforms, all constituting one signal. So the idea of ICA is that if we have enough subjects that have these signals that are composed of the same waveforms or target independent signals, we should be able to put them in the ICA machine, and they should be able to recover the original independent signals. And the reason for that is because if you look here, one signal is red, the other is blue, and it makes a purple signal, right? Red and blue, purple. Well, if you have a bunch of subjects that have different kinds of purple, you should be able to use those different kinds of purple to extract a red and a blue. Now, that is just an analogy for signals. Now, I'm taking a different approach called fusion ICA that asks the question, this waveform, like an N170 or a P300, which regions in the fMRI co-vary with these waveforms, so that we can look at a very precise slice into the fMRI using these components. And after about like 7 hours of ICA algorithm executing, we get this, the P3B load effect. So this is a group mean, and we identified this right here as the component because at the 300 millisecond mark, the positive deflection, this corresponds with the group mean positive deflection. And this is the corresponding covariant fMRI. Here, this is the group mean, the yellow, and then the blue, we identified that as the N170 component because it dips at 170 milliseconds, and it corresponds with that negative deflection. So this came from the occipital region, the O1 and the O2 electrodes. Cool. So remember, we expect to replicate an inverted U relationship at the maintenance period. And when we look at the fMRI map, that the only significant effect here was a inverted U effect in the prefrontal cortex. Fantastic. We also find in the N170 component 6 different significant effects, and they are all also inverted U relationships, so upside down U's, right? And these come from the frontal parietal network and the prefrontal cortex. The frontal parietal network known to have executive control over your working memory. So we did end up replicating the findings in the first hypothesis, and now in the second hypothesis, we expect to find linear relationships in the regions associated with cognitive control. We did not find a linear relationship between working memory capacity and load effect in the P3B component, but in the N170 component, we did find this. We found a significant linear relationship, positive linear relationship between capacity and load effect during the encoding period, the period in which they are trying to encode the faces. And we see these in the frontal eye field and the inferior frontal junction. The frontal eye field plays an important role in the control of visual attention and eye movements. It's basically responsible for your eyes scanning the faces. Its earliest activations happen within 45 milliseconds, so it's great that we did this fusion analysis because we wouldn't have been able to detect it otherwise. And this for me is interesting because it really demonstrates how having utmost control over your eyes processing and scanning over the images will result in better working memory capacity in the maintenance period. And then we see in the inferior frontal junction, in the left and the right, so right here and right here, to have this effect. And it is known to exhibit greater functional connectivity with the lateral occipital complex when working memory load increases, which would make sense because the lateral occipital complex is what's processing these images, these faces. So does this region have top control? Does it have top-down control over these regions? And then the last thing that we would want to ask is, sorry, we had a problem there. There we go. The last thing that we would want to ask is this connectivity, it could serve as a bridge between precision-related and load-related cortical regions. Now, how does that relate with the frontal eye field? So just to make sure that this was only something happening in the encoding period, we compared these regions to what was going on in the maintenance period. We did not see such effects, implying that these regions are really, really most helpful while you're encoding the information. So we actually ended up confirming both hypotheses with new, very interesting work ahead of us. So further analysis is we're going to compare my regions of interest with our previous lab findings. And finally, we will do post hoc tests on the ANOVAs, correlate accuracy with load effects, and examine the data from the retrieval period. I want to give a big thanks to the prop cohort and the Risman Memory Lab for sponsoring the research project and for amazing mentorship. I finally want to give a big shout out to my graduate mentor, Catherine, who just spent countless of hours debugging with me. Absolute, solidarity and just incredible mentorship. I don't know anything about fMRI or didn't know anything about it, and everything I know now is all thanks to her. And yeah, if you want to see an interesting discussion, come to the 10-minute talk I have at 1 p.m. on Friday.